Okay, so we are just waiting a minute more for everybody to join and then start the session. I can see 62 people so far has joined. So more people are just joining. Okay, so I think we can start now. Let me share my screen and we go from there. So hopefully everybody should see the share screen now. Okay, so hello everyone and welcome to this biomedical engineering event. This event is brought to you by the IET with the support of Andrew Caldos and Robert McDonald from Mercy and Western Cheshire Local Network. My name is Ashkan Eliasi and I am a Senior Research Associate in Biomechanical Engineering Group at the University of Liverpool, which is led by Professor Ahmed Al-Sheikh, who is one of our speakers tonight. I am also the Vice Chairman of Young Professional Group at IET Local Network and supporting the establishment of Biomedical Engineering Group. Today, myself and Dr. Andrew Caldos will host this session. We have 4 speakers from University of Liverpool. But before I introduce them to you, let me tell you a little bit about our local network. So the map is showing the area that we cover. So we have Cheshire here, Liverpool is there, Wigan up here, and we include Isle of Man. We have, so far, we have 6 different groups as part of our local network area group. This event is part of manufacturing and management group. Biomedical engineering group has not been established yet, it's in the progress, but for now it is under the same umbrella as this MM group. So the aim of the group is to share engineering success and innovation, to promote novel technologies, techniques, and best practice in the region, and to raise awareness of the importance of design, manufacturing, management, and their integration. We have various different activities. They include lectures, workshops, technical visits to recognition events, annual dinner. Everything that's happening in the local network is being published in our newsletter. And this newsletter is available in PDF. So if anybody is interested, you can have a copy by just emailing us. I provide the contact details of those who can help you with this at the end of this presentation. So the timetable for today, I'm providing the introduction, then we will have a talk by Professor Rachel Williams on tissue engineering in the eye. Then Professor Ahmed Al-Sheikh will talk about where we are after half a century development of ophthalmology. Then it will be followed by an interesting talk by Dr. Rochelle Dassa on nitric oxide to treat infections, go for no. And lastly, we learn about transforming healthcare with AI from diagnosis to prognosis by Dr. Yalin Zheng. Some housekeeping. When the speakers are delivering their presentations, please mute and stop your video. This session is being recorded and will be available online from IET website and YouTube. Use the chat not the chat actually, just use the Q&A button at the bottom of your screen to type your questions while the presentation is in progress. And the Q&A happens right after each presentation. So we have 4 speakers, each speaker has 20 minutes, and this 20 minutes includes the Q&A time. Dr. Andrew Caldos will kindly read selected questions to the speaker, or a speaker can read the questions themselves. If the time didn't allow us to answer your questions, please accept my apology and contact the speaker directly by email. So let me tell you about our speakers. Rachel Williams is a professor of ophthalmic bioengineering at the University of Liverpool with over 25 years of experience in the design and development of advanced materials for medical applications. Her expertise lies particularly in the design and characterization of the bulk and surface properties of material properties and how to modify them to optimize the properties for a specific application. She works closely with both clinical and industrial partners to ensure the appropriate translation of her research. She currently holds a fellowship from EPSRC on building advanced materials to treat vision loss under the Engineering for Growth Flowsheet scheme. Ahmed Al-Sheikh is a professor of biomedical engineering at the University of Liverpool and head of biomechanical engineering group. He is also a member of NIHR Biomedical Research Center for Ophthalmology based at Morpheus Eye Hospital in London and a member of Beijing Advanced Innovation Center for Biomedical Engineering based at Beihang University, Beijing. Professor Al-Sheikh's research includes characterization of material properties of ocular tissue, study of corneal topography, and development of medical devices to improve clinical practice in ophthalmology. Dr. Rochelle Dassa leads the antimicrobial biomaterials group at University of Liverpool. Her research focuses on the development of nitric oxide delivery platforms for treatment of multi-drug resistant infections to be used for skin, ocular, and bone tissue engineering applications. She has over £2 million in research funding with over £1.5 million as a PI. Dr. Dassa is passionate about supporting the next generation of scientists and has supervised students from various backgrounds to deliver new solutions for infection control and tissue engineering applications. I'd like to use this opportunity and congratulate Dr. Dassa for being awarded 600K from UKRI yesterday to develop a new antiviral coating for personal protective equipment to help stop the spread of COVID-19. Dr. Yalin Zheng is a leader in ophthalmic imaging at the Department of Eye and Vision Science, University of Liverpool, excuse me. He also has an honorary contract with the St. Paul's Eye Unit of the Royal Liverpool University Hospital for clinical research. He received his PhD in computer science from the School of Electronics and Computer Science and University of Southampton in University of Southampton at 2003. His main research interest is in the field of medical imaging, computer vision, and artificial intelligence. He has been a co-author of more than 150 papers in this field. So before I hand into our first speaker, I would like to tell you quickly about the future events of Manufacturing and Management Group. So on 3rd of February, we have Internet of Things for healthcare. On 10th of March, we have advances and applications in optical 3D metrology. And on 21st of April, we have a technical visit to Bilfinger, UK. On behalf of IET, I would like to encourage you to attend events, join a group, and involve your company. If you would like to receive a copy of the newsletter or get involved in our activities, please contact these people. We have Andrew Caldos, who is the chairman of Manufacturing and Management Group. Michael Gilbert is the deputy chairman of the group. Rob McDonald is the immediate local network chairman, and Godfrey Evans, who is the current local network chairman. If you have any comments, feedback, or inquiries about this event, or interested to join the biomedical engineering group, please feel free to contact me via social media by scanning the QR code now, either from Twitter or LinkedIn, or alternatively, email me at a later time. So without further ado, I should announce that we have 93 attendees so far. Without further ado, I would like to invite our first speaker to share her screen and join the call. Professor Rachel Williams.
Thank you, Ashkan. Do you want to stop sharing your screen and then I can share mine?
Yeah, sure. Okay, so you should be able to share.
Thank you very much. And it's a great pleasure to be speaking to you today from my computer. I'm going to introduce you to some of the work that we've been doing in relation to tissue engineering solutions for eye diseases. I don't wish to teach people to on something that's too easy for them, but I thought it might just be easy to start by looking at the eye and what some of the considerations are that are important in terms of looking for solutions for sight loss. So obviously in an eye, you have your eye here is the front of your eye. This is called the cornea, and light comes in through the cornea and across the anterior chamber here. And then this is your iris with the pupil in the middle, and the light goes through the pupil and into your lens, and it goes through the lens across the posterior chamber of your eye, which is full of vitreous, a jelly-like substance, and it then hits the retina, which lines the back of your eye here. And your retina is a neural tissue, and that stimulation of the neural tissue then sends signals to your brain via the optic nerve. So in a very simple process, if an eye is going to work and the vision's actually going to, and you're going to have vision, the light has to be able to get into your eye through the cornea. It has to pass along this light path. It has to get to the retina, and the retina has to work and pass the information down your optic nerve. And the problem that can occur in sight loss is that any part of this light path, if you like, can be damaged or can be interrupted, and then that causes a problem. So for example, you could have a problem with your cornea, and so the light can't get into the cornea, and the rest of the eye could be working perfectly well, but you wouldn't be able to have proper vision. Alternatively, the light can get in through the cornea, but your retina isn't working or doesn't work properly, and then you can't, doesn't matter how good the light is at getting into your eye, if the retina can't pass those messages to your brain, then also you'll have sight loss, you'll have problem with your vision. So the two particular examples that I want to talk to you today is one related to the cornea and one related to the retina. Right here at the back of the retina by the macula, there's a condition called age-related macular degeneration, which many of you may have heard of, and some work that we're doing on trying to see if we can find a solution to that problem. So if we then look at the other aspect and look at a very simple stage of what do we mean by tissue engineering? And what I mean by tissue engineering is that we take a material, which we call a scaffold, and we can build this material. And then we take cells, and those cells come from a biological tissue within a particular body. We add the cells to the scaffold, and then we produce what we call a construct, a tissue engineering construct, where the cells become embedded or involved with that tissue, that synthetic material. And if we look at this from a very simple point of view, clearly we've got to design the scaffold, and we've got to make it so that that scaffold interacts in the correct way with the cells. We need to choose the cells, so there are many different cells within the body, and we may want to choose cells that come from a particular patient themselves and do an autologous graft, or we might actually need to use cells from another patient if the patient's own cells are very degraded and not working in the way that we want them to, and then do an allograft, so you actually take cells from somebody else. What we have to do, though, is make sure that depending on the process that we need, that both the right cells and the right scaffold are chosen and designed so that the response we get at this stage is able to do the job that we want that tissue engineered tissue to do. And it doesn't necessarily mean that needs to recreate exactly the tissue that you're replacing. We need to look at what the job is that construct needs to do and make sure that it's going to do it, and not do it just for a short time, but do it for the length of time that's required for that particular, to solve that particular problem. So from an engineering point of view, we've got to actually make this whole process work. It's not a question of making one or other bit work and hoping that the other bit comes along and sorts itself out. So we need to look at it in its entirety. So if we look at the cornea, so this is one of the areas that I'm particularly interested in. So the cornea is right here at the front of the eye. And this is here, you can see, we talk about it as being the window into the eye. So this is completely transparent, and this needs to maintain transparency so that the light can get into the eye. But the cornea has an interesting structure. So if we look at it as a material, it's a structure that has this linear or laminar, if you like, of collagen structure. And within that collagen structure, there are a number of cells. And these are our stromal cells. So these are cells that have a very specific property, and they're fibroblastic-like cells. On the top of the cornea, you have the epithelium, and this is a stratified epithelium. So you've got multiple layers of epithelial cells. And this is rather like the skin. So this is your protective barrier that's making sure that things don't get into the eye that you don't want to get into the eye. And on the inside layer of the cornea, so this is the inside of your eye, you've got a monolayer of these endothelial cells. And these endothelial cells have a very specific function, which is to pump water out of the cornea, the stroma of the cornea, to keep it at the right level of hydration. And that's what keeps it transparent. And if these become damaged, they have very poor proliferative potential, very, very bad at being replaced in terms of a healing reaction. And as these become damaged, then they can't pump properly, and therefore the cornea swells. So this is the one area that I want to discuss where we've been doing some work, where you get endothelial dysfunction. So a normal corneal endothelium, if we look at these cells on fuss, if you like, we always talk about this as being a cobblestone-like pattern. So these are the monolayer cells, tightly packed as a single layer. And these are if they're like this and healthy, then they can pump the water out of the eye and keep the hydration levels of the cornea and keep the hydration levels. And in that way, you end up with a nice transparent cornea. You have conditions such as Fuchs endothelial dystrophy, where those corneal endothelial cells become damaged. And these are areas where there are no cells. So the cells have become degenerate and have died. And this is a layer of cells that then is unable to pump properly, and therefore the cornea swells, and when it swells, it loses its transparency. Currently, the only way to treat this condition is to use a corneal transplant. And that's a corneal transplant is when somebody who has passed away, they donate their eyes for transplant, and then those cells, this layer of cells plus a small amount of the end of the stroma is then taken by the surgeon and transplanted into a patient's eye. And in many ways, that's a very successful procedure, but there's a shortage of corneal transplants available for people, and for some people, it's not always successful. So we hypothesized that there might be a way in which we could take one corneal transplant, we could design a scaffold made of a particular material that we could then expand these cells on in culture, such that we ended up with a monolayer of cells on a number of different scaffold materials, so that they would, you could take one donor cornea and expand it so you might be able to treat, say, 4 or maybe even 6 patients from one cornea. And the idea would be is once you'd got the cells growing onto the scaffold, you would then come and the surgeon would be able to transplant that gel plus the scaffold, plus the cells back into a patient and recreate a very nice corneal endothelium without having to use one donor for every single patient. So to be able to do this, we needed to design a scaffold that was going to allow us to expand these cells. So the work that I've done on this is using a novel peptide hydrogel. So this is made out of polyethylene lysine. This is a readily available peptide that's actually used as a food preservative. So you can buy it very, very simply. It's a very simple peptide structure. So this is its structure here. So you've got one peptide joined to another peptide, sorry, one amino acid joined to another amino acid, joined to another amino acid, but they're all the same amino acid, which is lysine, polyethylene lysine. What we do is we take the polyethylene lysine and we cross-link it with dicarboxylic acids. So you can form an amide bond like this between the carboxylic group on the acid and the amine functionality on the peptide. And the really nice thing about this family of materials is we can use different diacids, so they have different lengths. We can use different densities of polymer, so different concentrations, if you like, of the polyethylene lysine. And we can use different cross-link densities. So we can cross-link it at 90% or 70% or 30%. And by cross-linking or changing all these different properties, we can get a family of gels, which we can then start to tailor for the particular properties that we want. But because it's a peptide, it models the extracellular matrix, protein-like matrix that you would have in the cornea naturally. And therefore, it's really likely to have good properties for the cells to interact with. It also has a high water content, which again is good for replacing tissue. So if you can see here, by changing those properties, the density and the cross-linking, you can see that we can change the mechanical properties of this material. And just show this again, that I've just made this blue so you can see it, but you can see the sort of properties that you can get. So although this is a peptide, which is a very delicate gel-like material, we can actually get something here that's handleable, which the surgeons then can actually use to use the surgery, which is obviously very important to be able to get it back into the eye. The other nice thing about this material is it has extremely good transparency. We can control the transparency again. If the diacid becomes too long, we start to lose the transparency. But you can see here from this little video that when we put it into water, you can basically not see the gel. It's got such good transparency in terms of the way it interacts with the light. So this material seemed like a good place to start, but obviously it's no good unless we can get the cells to grow on it. So we did a number of studies where we took corneal endothelial cells, actually from pig eyes, because we can get pig eyes very easily from the local abattoir, and we can then grow these cells. And this is the monolayer cells growing onto our hydrogel, our peptide hydrogel, after 3 weeks or after 5 weeks. And then here we've stained these cells with a particular stain that demonstrates that they have a pumping function. So it's not just that the cells are there and growing on it, they're actually functionally active within this opportunity. So what we've also then done is we've taken this gel and we've implanted it into a pig eye. It is ex vivo, it's just a pig eye that we've got from the abattoir, but our clinical colleagues that we work with of using this surgical equipment that they use between corneal transplants, they have demonstrated that they can do a mock surgery, and this is the gel here sitting inside the cornea of an eye that's been analysed using OCT, which Yalin might talk about a little bit later in the talk. But you can see here that when we can then measure the thickness of this, and if we have done a few studies which have demonstrated that when we do this, we can maintain the thinness of the cornea, so the cells are actually pumping within our model. So we're very encouraged that this is an opportunity where we've designed a material that the cells can grow on. We can design it with the right properties so that it can be used surgically. It has very nice transparency properties, and we are taking this forward now with some colleagues to see if we can get it forward into preclinical studies so that we might be able to take this forward to a clinical trial maybe in a few years' time. One of the other things about this gel is we can make it into a porous structure rather than just a sheet, if you like. And then if we do that, can we actually get the stromal cells also to grow into, migrate into this? Because we know that we can get this endothelial layer growing on it. We actually also know, although I'm not going to show you the data today, that we can get epithelial cells growing on it. Can we also get the stromal cells? You can see here we've created, this is our cast material, which has a very smooth surface looking under the SEM. But if we create a fragmented gel, you can see that we have a really nice porous structure from the surface of the gel. We can grow our endothelial cells on this fragmented surface, but this shows where we've seeded stromal cells onto this. This is the top surface, and then we've looked at various different depths into the gel, up to 150 microns, and you can see that these cells, having been seeded on the top, have migrated down into this surface, and they have this very nice fibroblastic morphology as you would expect them to have, right down into the depth of the material. So we're confident that we, in a laboratory sense, in the laboratory, in a dish, we can get stromal cells, epithelial cells, and endothelial cells all growing on a material like such as our peptide hydrogel, which is an encouraging step moving forward to trying to create potentially a synthetic cornea incomplete. This work is obviously, as I say, very much still in the laboratory, and it's going to take a long time to translate into clinical practice, but it is an encouraging step that we're at the moment. So the other area I just wanted to talk about was a new treatment for age-related macular degeneration. So this is a problem where we have a problem at the back of the eye here underneath the retina. Age-related macular degeneration is the commonest cause of vision loss in the over 65s in the developed world. And as we're all getting older and our populations are getting older, the numbers of people suffering from age-related macular degeneration is increasing. It's a disease associated with a damage underneath the retina. So this little model here, schematic, this is the retina, which is your photoreceptors. And then you have a layer of retinal pigment epithelial cells, and it's a damage to these cells that causes the loss of vision within this region. And basically what happens is you begin to lose your central vision. And this vision, just this central spot, tends to progressively get larger. So we've hypothesized that if we could replace this layer, here's your layer of retinal pigment epithelial cells sitting underneath your photoreceptors of the retina, and underneath that is a choroid, the blood vessels of the choroid. If we could design a substrate that we could grow these retinal pigment epithelial cells on, and take that and implant it back into the eye underneath the retina, could we then replace the function of the damaged cells with healthy cells? So again, it's back to designing the material of the substrate to grow the cells on, and to choose which cells we're going to use to be able to do that. This work, we've worked using polytetrafluoroethylene or expanded polytetrafluoroethylene. You all know this is Gore-Tex. You're all very familiar with this material. What you may not know is that this is what Gore-Tex looks like underneath a scanning electron microscope. Now, it's nice, it's porous. We need it to be porous because we need to be able to get nutrients and waste through this to the cells. It's available in very thin films, and it has reasonable mechanical properties. But as you all know, Gore-Tex is very hydrophobic. That's why it makes such good waterproof material. And as such, because it's hydrophobic in comparison to the hydrogels we were talking about, the peptide hydrogels, it's not very good for cell adhesion as it is, because cells like to spread on hydrophilic surfaces rather than hydrophobic surfaces. So what we've done is, over the years, is we've developed a way to modify the surface with a gas plasma treatment. So if we look at this, what's happening here, here's our chemical formula for Gore-Tex. You've got a very nice fluorocarbon. If you put a drop of water on that, you get a hydrophobic surface as you would expect. So a really nice beaded up drop. If we treat it with a plasma, and we use an ammonia gas plasma here, this is sufficient energy to break some of these carbon-fluorine bonds. We take it out and put it directly into water, and we get hydroxyl groups incorporated into that surface. And as you can see here, when we've done this, we get a very nice hydrophilic surface. And all we've done, because it's just a surface treatment, is we've modified the surface without affecting the bulk properties of the Gore-Tex. Just to show again, hydrophobic at the beginning, and then hydrophilic after we've treated it. So what we've then done is we've demonstrated if we take human retinal pigment epithelial cells, and if we grow them on the untreated surface, these cells stay beaded up, and they really don't like to spread on it, so they just stay rounded as similarly to the drop of water. But if we take the same cells and put it onto the ammonia plasma treated surface, these cells, rather like the corneal endothelial cells, create this cobblestone-like pattern. And these cells are how we need them to be form the monolayer that's going to be implanted back under the macula to try and regain sight within the retina. So we have a material now which is very good in terms of allowing the cells to attach and grow in the way that we want it to. We have to choose the right cells, and there are a number of different groups around the world working on this as a potential strategy for treating age-related macular degeneration.
Rachel, I'm sorry to interrupt. We just have 1 minute left.
Perfect, I'm nearly finished. So in this instance, what we want to do is to choose to use iris pigment epithelial cells. So these have been taken from the iris of the patient that this treatment is going to be used on. So we can take a biopsy from the iris, we can expand those cells in culture, and the eye beam, we then grow them onto our substrate, and that substrate should be put back into the eye. And one of the things we're working on at the moment is the instruments to be able to do this process. So in summary, just to bring that to a rapid conclusion, these are the two areas that I wanted to introduce you to: the potential for corneal reconstruction to replace corneal transplants using a peptide hydrogel and donor cells from the corneal donor, or alternatively a subretinal transplant where we're for treating age-related macular degeneration. And in this case, we use a very biostable synthetic material, which has a lot of experience in use in medical devices, but we need to surface modify it so that the cells can grow on it. And our approach is to use autologous RPE cells, whereas other people are looking down the stem cell route. And just to finish then, to acknowledge that obviously I don't do all this work myself. I have very wonderful teams of postdocs and PhD students who work with me, and also my clinical colleagues who are very helpful in trying to make me do this process and come up with things that are clinically relevant. And of course, I've been very fortunate to be funded by many different charities and research councils. And I thank you very much for listening. I'll stop sharing now, and I'm very happy to answer some questions.
Thank you very much, Hazel. I think, Rachel, I'm alive now. I think so. Is it okay?
Yes,
can you hear me? Can you hear me?
Yes, we can hear you.
Right. We have thank you very much for this very interesting talk. I really enjoyed that. I have three questions now. I wonder if you have time for that. The first one is a good one. Can you harvest good cells from the patient's cornea instead of using a donor cornea? Would this be helpful? Is there a rejection problem with donated corneas?
That's a very good question. In terms of the donor corneas are well accepted as a transplant tissue, and rarely cause a huge problem in terms of rejection. It's one of the areas where it's actually been very, very successful. Using the own patient's own cells, in the terms of corneal endothelial cells, that's probably very difficult because we don't they don't grow very well. And if you've got a patient that's got damaged corneal endothelial cells, you're very unlikely to have enough to be able to harvest their own cells. In terms of the corneal epithelial, that is sometimes the case, and you can sometimes take the cells from one eye, using the other eye if you've got damage to just one of your eyes. So it does depend a little bit on where the cell, what your problem is and what cells it is that you're trying to replace.
Okay, thank you very much for that. The second one is, have you tried growing cells from other organs, specifically vascular cells, on this construct?
So a lot of people are growing vascular cells on ePTFEs. I'm sure maybe you're aware from your question. So the one of the materials that is used in vascular grafts, there's still a problem with a vascular graft, even with Gore-Tex ones, when you get very small. But it is possible to grow other cells on this material. We haven't really done a lot of work at ourselves, but we've particularly aimed our work really at the eye.
I see, right.
I'm sorry, Andy, to interrupt. I think we have to move on to the...
Thank you very much. I still have a few more questions here, but okay.
Yes, I
We
think
have no time for that, but
I might be able to type some answers to some of them in the chat.
That's all right. Okay, so we carry on with that. Back to you, Ashkan. Thank you
very much. And
thanks, Rachel. Thank you.
thank you. So I would like to now invite Professor Ahmed Al-Sheikh. Professor, if you could unmute yourself and share your screen, please.
Thank you, Ashkan. So can you see my screen now?
Yes, we can.
Okay. Right. So good evening, everyone, and welcome to this talk. My name is Ahmed Al-Sheikh. I'm a professor of biomedical engineering from the University of Liverpool. It's actually a great pleasure to be here, although I would have preferred very much to talk to you in person and to see you and to shake some hands at the end. But in any way, so this talk is about a historical journey of corneal biomechanics. I want to show you, just explain to you from what I see, how corneal biomechanics have developed in the past, and then this hopefully leading us to a very exciting story over the last 20 years. I think you will be aware that biomechanics is part of structural mechanics. Structural mechanics has seen steady progress that has been documented for at least 5,000 years, with very known, very good examples. However, the last 4 centuries, probably from the 1600s onwards, we have seen an incredible progress, led by very well-known names like Euler and Newton and Bernoulli, and these have introduced concepts that have become an embedded part of our life, an integral part of our life, like elasticity and stiffness and tissue mechanics and so on. In biomechanics itself, biomechanics is basically structural mechanics when it's applied to biological tissue. If you look at historical examples, you will actually find very little. So while structural mechanics, in terms of civil and mechanical engineering, has been developing fast, biomechanics has really lagged behind. One of the first major works was publications by Leonardo da Vinci in the 15th century. And in this work, the focus was on the musculoskeletal systems. So this was bones, muscles, cartilages, tendons, and it really focused on how the body maintained its equilibrium, maintained its stability, managed to move, carry weight, and so on. And this focus on the musculoskeletal system persisted for a very long time, and I would say even until now, that you will find that a big part of the biomechanics work is still focused on the musculoskeletal systems. And the biomechanics of soft tissue in general received much less attention. If you now jump to the 21st century, you will find some examples of biomechanical applications in soft tissue in the cardiovascular system. And here you have 2 examples that I think everyone will be aware of: the stents and the balloons used to widen narrow vessels, blood vessels. These have been really great inventions and saved lots of lives. And while this has been going on, work in other areas, including ophthalmology, is still lagged behind. So I'm going to concentrate now on corneal biomechanics, and I'm going to take you to a conference. It's the ARVO conference, the Association for Research in Vision and Ophthalmology. This is the biggest international conference in ophthalmology that's held in the US every year. And in 2003, they had a keynote speech on corneal biomechanics, which I thought was very, very unusual. And actually the speaker then talked about corneal biomechanics being in the Middle Ages compared to other fields of biomechanics. And actually, a couple of years before that, there was a very interesting publication with this really interesting title, "The cornea is not a piece of plastic." And that was published in 2000, in the year 2000, about 20 years ago. And if you read this paper, you will find it really amazing, because it's something like secondary school level introduction of biomechanics to clinicians. And at the time, biomechanics was something, it was a very novel concept. Very few people actually understood what biomechanics was about. For many people, it was just colorful images of numerical modeling of the cornea and the sclera. At the time, that was the time when I started work in ocular biomechanics. And one thing that I did at the time was to look at the literature and actually collect together the stress-strain behavior, the mechanical behavior of human tissue that was tested experimentally. And I was really horrified to look at this, because here you get the stiffness of the same tissue tested in different labs and coming up with stiffness differences of up to 40 times. And I used to tell my students that this is like designing a bridge and telling the client that the formation of the bridge is somewhere between 1 inch and 1 meter. And this was experimental behavior, which means that it was dead tissue, ex vivo tissue. So you can imagine how it was testing live tissue. So why is biomechanics important? And why are we interested in this work? Actually, there are lots of applications about disease progression and treatment of disease. These applications interact or interfere mechanically with the cornea. And unfortunately, if we don't have the ability to measure corneal stiffness, there is no way that we can optimize these applications. And this is why, you know, you find things like in this example here, the cross-linking treatments. So this cross-linking treatment is really important, because there are lots of people who are suffering from a disease called the keratoconus, which is softening of the cornea. And if you want to stop the progression of this disease, you stiffen the cornea using cross-linking treatment, basically using UVA treatment. And the decision here is to treat or not to treat. It is not how to treat. It is not the intensity or the duration of the treatment. It's treat or not to treat. And this is basically simply because we cannot, or we could not until recently, measure corneal stiffness in vivo. The first ray of hope came in, I think that was in 2005, 2006. And that was the ocular response analyzer produced by Reichert in the US. And it's a machine that applies an air puff, air pressure, air jet to the cornea and looks at the deformation of the cornea as a result. And they took the deformation of the cornea, analyzed that, and came up with two parameters, one called CH, corneal hysteresis, and another one called CRF, corneal resistance factor. And they said that these two parameters are related to engineering biomechanics. The problem we have is that these two parameters are not related to the engineering stiffness parameters that we know. For example, they are not related to the tangent modulus. They are not related to the viscoelasticity parameters. And therefore, we have no idea what they mean. And people did not have any idea what they mean. And actually at the bottom here, this is a very brief extract of a paper that I have seen a couple of days ago, and it shows you that, you know, it's just read the studies reported higher CH in one study with a certain disease, both higher CH and higher CRF in one study, higher CH accompanied by no change in CRF, that's another study, no change in both CH and CRF, that's a fifth study, and then increases in CH and decreases in CRF. So basically, these two parameters are absolutely all over the place, and we don't know what they mean. A more recent example was the Corvus ST, and the Corvus was introduced in the market in 2013, 2012. And the Corvus is based on a simple, on a very similar concept, similar to the ocular response analyzer, but it differed mainly in the amount of information it collects on corneal behavior, corneal deformation. So these deformation parameters, some of them you see on the right of the screen, they were called the dynamic corneal response parameters, or the DCRs, and these parameters were still unrelated to engineering stiffness parameters, and therefore, they suffered again the same problems as CH and CRF. For us in Liverpool, this was a really exciting challenge. It's something that people have been trying to sort out for a long time, and we thought that this would be a really good area to work in. So we used a combination of experimental testing, numerical simulation, and clinical validation to come up with a parameter called the stress strain index. So what is a stress strain index? Stress strain index is intended to give you the full stress strain behavior of corneal tissue. There is a trick here because, as you know, the stress strain behavior of biological tissue is nonlinear, and therefore you need at least two parameters to define this curve. If you are interested in knowing how we managed to use one parameter to define the whole curve, I can explain this to you. But basically, the stress strain parameter, the stress strain index, is intended to give you the whole stress strain curve, and therefore the tangent modulus at any stress or strain. And because it's a material parameter, it is independent of corneal geometry, and it is independent of the eye's internal pressure. We have done a validation studies on the SSI, and this is one result. So I mentioned to you before that keratoconus is a disease that causes softening of the tissue, and this is a result that shows you that for healthy people, this was the average SSI, and as the disease became progressively, as keratoconus became progressively more advanced, the SSI value decreased, indicating that the cornea was becoming softer, as you would expect. And then we did another test for people who underwent the cross-linking treatment that I mentioned before. So this is cross-linking treatment used to stiffen corneal tissue, and you would expect that the tissue would become stiffer at the end. And the problem at the moment is that there's no optimization on one hand, but on the other hand, there's also no way of telling that the treatment has worked. So when we use the SSI on a population that we got from database from Liverpool here, we found that in 92% of the cases, the stiffness has increased. This is another really exciting development that we have, in that the SSI, as I explained so far, is a single parameter representing the mechanical stiffness of the whole cornea. However, through or based on the links that we know exist between the microstructure of the corneal tissue and the biomechanical behavior of the cornea as an organ, we can actually convert the single reading of SSI into a map of SSI. And we have done this, and I'm showing you here two examples, one of a healthy cornea, and the other one is of a keratoconic cornea. And in the keratoconic cornea, you see clearly that this area here of low softening, and it's not only that, but also know that it is here, and we know the degree of softening that this area has suffered, and therefore, if in the future you want to optimize the cross-linking treatment, you can concentrate the cross-linking agent on this region. So, overall, where are we? So I think corneal biomechanics has a lot of catching up to do. It started late, and therefore there is a lot of work that needs to be done, and therefore this is a really good area of research to work in. My expectation for the future is that I expect that corneal biomechanics, recognition of the importance of corneal biomechanics will steadily grow, that it will form an integral part of clinical practice. I expect that it will be measured as a standard procedure, and that the information provided on corneal biomechanics will become increasingly detailed, and that it will be used to optimize treatments, as I mentioned before. And I leave you with a group, with a picture of our group that was 3 years ago. Thank you.
Thank you very much, Professor. So I would like to just ask Andy to.
Yes. Okay, it's a couple of questions here.
Sorry, I think the questions are for Rachel.
I was going to say that. So all these questions are to Rachel. But anyway, it's thank you very much, Ahmed, for this presentation. It was really excellent. And all these cornea works, but I'm missing your labs. It would have been so nice to be shown around, to see all these measurements, what you do, what you've done in live, in live situation. But we can't do it right now. Maybe another sort of, in the future, I believe it's going to be a future sometimes. A kind of video clip would be so nice to show, because, well, all these things are difficult to catch sometimes. Well, let me ask one single question on the SSI maps. I just lost. What does it stand for, SSI? I lost that thing. Could you explain that to me, please?
Yeah. So Andrew, the so corneal tissue, like any biological tissue, has nonlinear behavior. So it starts under low load or low pressure or low stress. It is quite soft, and then after that it becomes stiffer. So the tangent modulus, which is a measure of mechanical stiffness, is not constant. It's small at the beginning and then high as you increase the loading. SSI is intended to give you the whole curve, and therefore it gets over the difficulty of having to compare E-tangent, the tangent modulus, which doesn't have a constant value. So this means that if I have an SSI of 1 and someone else has an SSI of 1.2, it means that his cornea is 20% stiffer than my cornea at any load or any stress level. The idea of the map is that it converts the single value that you get from the methods into a distribution, so that you can tell that here are areas where you have high stiffness, and here are areas where you have low stiffness. So in your treatment, you can then target the areas with low stiffness, to rectify the damage that took place because of the disease.
Okay, thank you very much for this explanation. And another short question here. Well, he says, can I hear about the air-proof test in informing clinical decisions?
Yes.
Test informing clinical decisions. Maybe it's about the pressure test, is it?
Yeah. So for instance, so we know that healthy corneas will have an SSI value of around maybe between 0.8 and 1.2. Mm-hmm. If you have let's say an SSI of 0.5 or 0.6, it will be very risky for you to go and do refractive surgery, because refractive surgery involves removing tissue from the cornea and therefore making it softer. So if your cornea is already soft and you do this to it, then you run the risk of having problems, serious problems, when the cornea becomes destabilized after refractive surgery.
Okay, thank you so much, Ahmed. Appreciate it.
Thank you.
You're welcome.
Back to you, Ashkar, please.
Thank you, Andy. So now I would like to invite our next speaker, Rochelle. Rochelle, if you could kindly unmute yourself and share your screen.
Second.
Yes, we can hear you. Yes.
Hi, so my name is Rochelle Dessa, and thank you for the kind introduction earlier during the session, and thank you for the invitation for me to speak a little bit about what we do in our lab in Liverpool here for you today. So as Ashkan had said initially, in our lab, we work with nitric oxide to be able to treat infections for the skin, eye, and bone. And I'll take you through a bit of an introduction about antibiotic resistant infections and things like that. And we'll continue to talk about how we try in our lab to to treat some of these infections. So probably pre-pandemic, the headlines like this were more prevalent and were maybe more shocking as well, where the world is essentially entering a post-antibiotic era in which infections that were once treatable will once again kill. That's because antibiotics are becoming less and less effective because bacteria are becoming more resistant. And the O'Neill report a couple of years ago has stated that by 2050, it is predicted that antibiotics will become obsolete, and this will result in about 10 million deaths per year, which supersedes cancer as the #1 cause per death. And in essence, what this means is that we're going to go back to the early 20th century, where infectious disease was the most prevalent cause for a decrease in life expectancy. And, you know, all of this was quite prevalent in the news, and the fact that antimicrobial resistance is a huge problem was very prevalent in the news. But with COVID taking over, it's kind of, you know, put this to the back burner. But what we all have to think about is that antimicrobial resistance is actually quite a slow moving pandemic, and it is to become a significantly bigger problem in the near future that needs tackling very, very efficiently by scientists around the world. So headlines like this, you know, will continue to plague us, where we see the term superbug that is, you know, that's thrown out there. And a superbug is something, a bacteria that's resistant to quite a lot of different antibiotics. The one headline that caught my attention a couple of years ago was that there was a woman in Las Vegas who was killed by a superbug that was resistant to all 26 of the antibiotics available in the US. And you know, if you think about it, most people don't think about having a common infection such as urinary tract infection or things like that that can escalate quite significantly. But if we run out of antibiotics and if we run out of ways to treat this, we will enter the medical dark ages again. So, sorry. There we go, sorry. So if we look at the antibiotic discovery pipeline, the first antibiotic was discovered in 1928 by Alexander Fleming, and this was penicillin. But if you can see, you know, just a little over a decade later, a resistance to this antibiotic was already discovered. And every single class of antibiotics that was discovered subsequently after sort of penicillins has had bacteria that have developed resistance mechanisms to these antibiotics. And if you fast forward to sort of where we are today, 2020, you can see that in the last 30 to 40 years, there have been no new antibiotics introduced at all. And part of the reason for this is that the pharmaceutical industry is not really interested in developing antibiotics at all. What they're interested in is developing drugs that you can take for a long time, such as like cholesterol drugs or blood pressure medication. These are drugs that will make them a huge profit margin. When you consider things like antibiotics, you take them for a week, 2 weeks, and the bacteria can develop resistance to these antibiotics. So it's actually quite a non-profitable industry. And so that's part of the reason why we have no discovery of new antibiotics that are out there. So if we look at bacteria, so let's take a step back and actually talk about bacteria. So bacteria are all around us, and they exist very symbiotically within our bodies. If you consider how many bacteria actually reside in your body, cell for cell, we actually are about 43% bacterial cells compared to human mammalian cells. All of these bacteria that live within us are mainly good bacteria, and they're not passive riders in us. They do things like they cover us in an invisible body armor. They help us digest foods. They help us make vitamins. They educate our immune system to keep bad bacteria out. So when we want to be able to kill off infection, when we want to be able to kill off bad bacteria, we have to also consider the good bacteria and the symbiotic bacteria that live amongst us. And if you think about how many genes are within your body, bacterial genes actually you actually have 10,000 more bacterial genes than human genes within your body. So when you're trying to tackle infections, you have to be able to consider not wiping out any of the good bacteria within your body. Now, the bad bacteria, those are the ones that you want to be able to kill. These are things that cause things like ulcers or toxic shock or food poisoning. And these are essentially what gives lots of bacteria bad names. But once some of these bacteria colonize your body, if you take antibiotics, essentially it should be able to cure the infection. But because antibiotics are becoming more obsolete, we need to find new ways of tackling these types of infections. So what we do in our lab is we think about different ways in which we can tackle these infections without using antibiotics. And antibiotics, if you think about it, was discovered very serendipitously by Fleming in 1928, where mold was able to destroy the bacteria. And so it is a natural, it's a natural mechanism for how bacteria get killed. And so what we think in our lab is lots of different organisms have been evolving over centuries and millennia, and they've been evolving ways of being able to fight these bacteria and live with these bacteria. So let's look at the different ways which different organisms can interact with bacteria and are able to fight off infections. So if we look at actually a plant initially, one of the ways a lotus leaf can actually prevent bacteria from sticking to it, which is really cool, is that they have this super hydrophobic structure. And with the super hydrophobic structure, Rachel showed you a really lovely image of a contact angle earlier, where hydrophobic structures have these really rounded water droplets on them. And the way they work is that because water can't stick to it, it just flows off, and as it flows off, it pulls all the dirt and bacteria off from around it. The picture in the background is actually the Dublin Spire, and if you've been to the city centre of Dublin, you've seen the spire, and the surface of it is created by a hydrophobic structure or self-cleaning structure, where they assume that because it rains a lot in Dublin, they would get enough rain for it to be a self-cleaning structure. But unfortunately, it doesn't actually rain enough in Dublin to make it a self-cleaning structure, so they still have to clean it. Another way that nature has found to be able to fight bacteria are sharks. Sharks are have these really interesting textures on their skin that prevent bacteria from sticking to them. So they have these structures called riblets, and scientists have copied this type of topography. And by putting this type of topography on various medical devices, what happens is that bacteria, when they adhere onto the device, they can't communicate because the topography prevents the bacteria from forming colonies. And in this way, bacteria don't stick and they can't cause an infection. And lots of people around the world are trying to copy this method of preventing infections on top of devices. Another really interesting method of fighting infections is from the Komodo dragon. The Komodo dragon is a creature that lives in a very sort of harsh environment that not many creatures can live in, and it is able to fight off lots of different bacteria. And what scientists have found is that the blood and the saliva of Komodo dragons have lots of these antimicrobial compounds, antimicrobial peptides that can fight off lots of resistant bacteria such as MRSA. And so it was just in the news actually a couple of days ago that I saw that scientists have been able to isolate some of these compounds and develop a new drug out of from Komodo dragons. Honey is another type of thing that a lot of scientists are looking at to be able to derive antimicrobial compounds. Certain types of honey have naturally antimicrobial compounds in there, and there's lots of scientists working trying to develop new drugs from honey. So within our lab, we thought, well, instead of looking at lots of these different animals and organisms, what does our own human body do to be able to fight off infections? And we have lots of different systems where we can fight off infections, but one of the things our body does is it produces this molecule called nitric oxide. Now, nitric oxide has lots of different functions in the body. It is antimicrobial, but it also is involved with things like cell proliferation, wound repair, immune response, cardiovascular homeostasis, neurotransmission, apoptosis, and angiogenesis. But the one we are interested in is it can kill off bacteria, and it actually, as a drug, it doesn't cause resistance. So we started getting really interested in using this molecule, and can we deliver this molecule to kill off an infection without causing resistance mechanisms in bacteria. So so how nitric oxide works is it combines with reactive oxygen species that are present within your body. It creates reactive nitrogen species, and these reactive nitrogen species will then damage different parts of the cell. It'll damage them of the bacterial cell. It'll damage the DNA, proteins, lipids, and it'll inhibit enzyme functions. And because it is part of your own body system of being able to fight off an infection, we have developed ways of not being affected by nitric oxide in certain concentrations. So it can target bacterial cells more specifically than eukaryotic cells. And so that is a really good thing about nitric oxide and being able to treat infections. So within our lab, we look at being able to develop nitric oxide donor platform technologies. We look at nitric oxide for lots of different applications, and I'll just run through really quickly a couple of the applications that we have within our lab. So the first one I'll talk about is the gels. So this is some of the work we've done actually in combination with Rachel Williams. She had talked about the polyethylene lysine gels that she has developed in her lab, and we collaborated with her to be able to tag nitric oxide onto the gels she has developed. So the application here is to develop antimicrobial contact lens bandages for the treatment of microbial keratitis. Microbial keratitis is a serious infection of the cornea, which can cause loss of vision. And one of the most common sources of getting this type of infection is because of inappropriate contact lens wear. If you don't clean your contact lenses, if you leave it overnight, it can result in this type of infection. You can also get it from trauma to the eye. And so once you get this infection, it is very difficult to treat with things like eye drops. So if you have this contact lens bandage that Rachel has developed, and you can put it over the eye, and we can deliver the drug nitric oxide to the infection, you can clear the infection without causing resistant bacteria. And Jenny in my lab has done most of the work in developing nitric oxide releasing contact lens bandages, and these bandages compare pretty well to ones that have been soaked with penicillin antibiotics. The second application that we look at nitric oxide releasing surfaces for are for things like developing antimicrobial wound dressings. So infection is one of the main causes of delayed wound healing. So if you have surgical wounds, trauma, burn wounds, chronic ulcers, one of the reasons that these type of wounds don't heal is because of bacteria that colonize the wound and form biofilms. But if you can treat the infection, you can perhaps kickstart the wound healing to begin again. And so what we do is we use a technique called electrospinning, where we spin fibers of bandages, and we then tag our nitric oxide molecule onto these fibrous mats to be able to release nitric oxide in a controlled and sustained manner, so that we can kill off an infection on a skin wound. So Miley in my lab has been developing lots of these different bandages by spinning different types of polymers together and by attaching nitric oxide to be able to kill off skin infections. So the second type of application that I will talk to you about today is for bones. So we started looking at titanium implants. So we looked at polymers and gels before, but we also wanted to see, can we attach nitric oxide onto metallic implants that are normally used for sort of bone tissue engineering applications. And the reason we wanted to tackle bone infections is because orthopedic implants, so you have things like fracture fixation, a joint replacement, hip and knee replacement, and you have different rates of failure for these type of implants. And one of the reasons for failure for these implants is because you have bacteria that colonize the surface, and once bacteria colonize the surface, the implant is essentially rejected, and you have to have a revision surgery to remove the implant. So if you can tackle the infection at the site of the infection rather than through a systemic antibiotic, it will make a massive difference for having the implant be retained into the body and prevent a revision surgery. So we looked at, so Miley again here looked at titanium surfaces, and she developed these different linker molecules which she could attach to titanium that had different abilities to release different payloads of nitric oxide at different rates. And what she wanted to essentially do is be able to release nitric oxide so that it would not kill off any of the osteoblasts or bone cells that wanted to adhere onto the surface, but it would kill off any of the infection that would have attached to the surface. So she did quite a lot of work, and what she found was that she was able to develop these type of implants where you were able to kill off bacteria on a few of her coatings, and these two coatings were also able to maintain osteoblast cell growth. So we were able to selectively damage bacterial cell populations, but not mammalian cell populations. So what we do in our lab is essentially we want to be able to find ways in which nature has been able to find new drugs. And we don't know where the next new blockbuster drug will come from, but what we are exploring within our lab specifically is nitric oxide, because it is one way our body is able to deal with different types of infections. And we are trying to mimic how our body does it to be able to fight off infections without using antibiotics. And with that, I would just like to end, and I would like to thank my group, who I've just spoken a little bit about some of Jenny and Miley's research, but they're an amazing group who do quite a lot of really innovative work. And I would like to also thank our funding sources, which we could not accomplish any of this work without them. Thank you.
Thank you very much, Rachel. So I think we don't have Rachel, any questions. I don't know, Andy, if you have any questions yourself, or shall we proceed?
Oh yes, if I could.
Sure.
Can you hear me?
Yes.
That's great. This nitrogen oxide, it's a fantastic stuff. To what extent is being used by the NHS now?
Nitric oxide, so it is approved as an inhaled therapy right now for neonates. I think it's approved in the US. I'm not sure if it's approved in the UK, but so that's its medical use. It's being explored within scientific labs quite a bit, but for you to be able to get a drug from sort of bench to clinic is a very, very long medical sort of like timeline for it to be able to succeed.
This convinced me that it has tremendous potentials in that.
Yeah, so it was discovered, like the potential of nitric oxide was discovered, yeah, in the 90s, and it was discovered, like it was used as a vasodilator. That was its first sort of, potential application, and people have been developing it for lots of different applications, and most recently antimicrobials.
Thank you very much, Michelle. Appreciate it. Thanks for your answer.
Very much. So
back to you, Ashkan.
Thank you, Andy. So Rochelle, if you could mute yourself.
Yeah.
And I would like to now invite Yalin, please, to unmute yourself and share your screen, if possible. Perfect.
Yeah, can you see?
Yes, yes.
Okay. Yeah, thanks, Ashkan, for the introduction. So I'm Yalin, and I'm going to talk about transforming healthcare with AI from diagnosis to prognosis. And so nowadays, AI is a very buzzword. And first of all, and this is my disclosure. And so today's presentation is my personal view, not any of these founders. And so AI is a hot topic nowadays. And so what are AI? So AI are a collection of technologies that make the computer or machines can and behavior or think like a human. So this is important and for many tasks, for example, planning, learning, reasoning, and problem solving, so on and so forth. And AI is not a new topic actually. And so it is coined in 1940s. So now it's about 80 years old. And then you can see there is a trend of acceleration of AI. And of course, there are golden age and winters. Because at one point, and the industry has a very high hope, and then AI cannot deliver what they want, then there is no investment. So there is winter. Luckily, nowadays, we are in another spring because of deep learning. So, and you might ask why deep learning makes difference. So here I use two diagrams to show the concept. So in a traditional neural network, you have input, and when you do a classification or diagnosis, and you need features to input this neural network and get the diagnosis. But where are the features from? So these have to be handcrafted and from a knowledge of experts, so on and so forth. And deep learning, for example, convolutional neural network, and is a new technique, and that can make an image, for example, as an input directly, and then through all the operations, it can make the prediction or classification. So the features are learned automatically in the process of prediction. So that's the difference. And so this comes successful because we have the data, and bear in mind, and garbage in, garbage out. So we need good data. And when we talk about the success of AI, we talk about, and no one can avoid talking about this big data set called ImageNet. It's over 1 million images, it's 1,000 categories. And so from 2010, there's a competition on machine learning program to see which program can perform best. And so in terms of classification and the localization of object in the images. And in 2012, there is a breakthrough. So, and this paper presents a very simple network. And nowadays, and you can see it only have 8 different layers from convolution and to pooling layers, and the input with the image and output with the prediction of 1,000 classes. And it has used 2 GPUs to make this work. And as a result, it produces the error, which is over 10 percentage points lower than the second and the best. So wow, people just shocked. And it marks the deep learning error. And also this is benefited from GPU. without that, actually 10 years ago. And Lecan and his team have used the convolution neural network, but cannot get it to work well because the limitation is availability of computing resources. They want more power to be able to do it. And nowadays you can see with deep learning from 2012, it make a big change. And then after 3 or 4 years, so deep learning can work better than human in this task. So that remind people, AI can be used for many tasks related to images. That's the reason why AI has become popular in healthcare. There are many things AI can do. And in particular here, I look at using images for detection and prediction and diagnosis. And so Rachel and others have set a solid background for clinical problems overall, and the eye is a transparent organ. So imaging is very, very important for the diagnosis and the management. So I'll show you some example by others. And first of all is a landmark paper published by Google in 2016. In this paper, they use color founders images. Those are the projection of the retina at the back of the eye. And they trained on 10 different networks to put an ensemble to detect referable diabetic retinopathy. Those are conditions of diabetes. And then they validated on 2 different data sets. And so their conclusion is that deep learning algorithm can produce a high sensitivity and specificity and for detecting diabetic retinopathy and macular edema. And this is very important. And the caveat of this study is that, and this is not a standard screening population to be bear in mind. And then in 2018, DeepMind and Moorfield have produced another paper. This paper is used OCT, optical coherence tomography, and to make a diagnosis of retinal diseases. OCT is similar to ultrasound, and it's using infrared light instead in order to get a high resolution cross-sectional images. And in this network actually, and first of all, they use deep learning to learn how to segment the features from these images, and then use these features together with the labels of each OCT images from over 14,000 and OCT images. They train different networks so that they can make a diagnosis. First of all, if the patient needs to refer or not, or the second is diagnosis of different conditions, and also to quantify the volumes. And so they showed this work, and they can perform the performance of AI can be as good as or better than expert, for example, from retinal specialist, optometrist. So the light blue region shows if anything outside that, and then it's worse than the AI model. And also, the other group, and led by Professor Abramov, has shown that, and AI actually can show performance for real clinic use. This is a study on 900 subjects, and they showed sensitivity and specificity better than and predefined endpoints. Those have been used evidence to get FDA approval for this first ever AI product. So this tells us AI nowadays is ready into clinical practice. So today I'm going to talk about the advantage of prognosis. And this is important to predict the future outcome of disease and or decide treatment, because this can help government to decide healthcare policy, and also this can help clinicians to decide the best treatment or early intervention for the patient in order to get best outcome. And in the past, this is not required for the clinician to do, and also it's based on their common sense or knowledge and experience. So now and future, can we do this by AI? Actually, and there are some recent work. Again, DeepMind and UCL Moorfield has developed a new tool to predict progression to wet age-related macular degeneration and using HCT images. So this is the diagram. First of all, and they have digital OCT images, and then they use their segmentation tools developed in the previous work, and then get the lesions, and then can use deep learning to predict the conversion of risk of AMD, and within 6 months of image. And also they have another branch to use image directly to make this prediction. So it's a combined model between image and lesion maps to make the prediction. And so this is and the journey of the patient. And so of course the patient have many visits. And unfortunately in their work, they only use one of the images to predict the next episode, if the patient will get AMD or not. So this is their results. And here is the ROC curve. And also the squares and circles show the performance of and the clinicians and all the combined results from different clinicians. And so in summary, and so because the prediction is not routinely required by clinicians, and the expert can still and perform better than random guess. However, and AI outperforms the majority of experts for prediction of a future conversion. This is very important, although the performance is not that good yet. If you look at, if you have a 20% false positive rate, then the sensitivity is about 50%. So this reminds us a similar problem. If for a patient, if you have many historical images, can we do a better guess with historical or longitudinal data? That's unfortunately in the literature, many work have worked on the prediction, but they only use a single time point. And so this is the only work and in the literature that uses OCT to make a prediction of conversion of AMD. And but similarly, they use traditional statistical method based on the extract features. For us, we want to do is if we have a longitudinal imaging, we want to make an end-to-end system that can make a prediction. This is to combine the strengths of deep learning and the traditional statistics models. So my students have worked on this big data set called the IRIS, and we use data from 4,903 eyes. And so about 10% of the eye have progression. And we split the data into training, validation, and testing. And first of all, we tested a standard method that is using a single image and use a deep learning network and to predict in the future and visit if that will be a conversion of AMD or not. And also, and this is the problem, and for if we use a single image, and also if we have multiple images, and how can we do it? So we borrow the idea of a gated recurrent unit. This is a deep learning network that can use different gates to control the flow of information so that we can combine information from different and images taken at different visits. So this is the architecture of the network. So we use a convolutional neural network to extract features from different images, and then we set a time window and try to compensate the different time intervals because of different and visit time between visits. And also we uses this GRU to combine the information, and then we make the prediction. So now it's the comparison results. So if we use single time point, 2 time point, or 3 time points, and you can see with using multiple visit points, we can get much improved performance in terms of AUC sensitivity, and also the specificity are increasing as well. So also show you the ROC curve. Basically, you want this curve as close to and the top left as possible. So you can see, and 3 points and 2 points are significantly better than using one single point. And so this is very important. We can achieve higher specificity and without losing sensitivity by using longitudinal image dataset. And so we also use the visualization techniques to visualize and what features the program can learn from this training process. They matched very well with the clinical features. And so the short conclusion here is, so as always, there are always dangers of predicting the future. And from our experience and others, so AI can help to make reliable and accurate predictions. If and only if we have enough data, GPU power, expert knowledge, and good algorithm. And so as always, AI is a new emerging thing. And there are so many challenges in terms of research, acceptance, ethics. For example, would you trust AI's prediction? And also how to deal with this discrimination possibility. If the AI predicts someone has a better, has a poor predicted outcome, would the insurance charge more? Is it fair? Those are the questions we have to bear in mind. So, but as always, we are confident AI will help improve or transform the healthcare landscape. And of course, this research is done not by myself only, and I have a good team to help with. And so all credit to them. And so I'm going to stop now and ready to answer any questions.
Thank you very much, Yalin. Andy, it's you now.
Can you hear me?
Yes.
Yes.
It's all right. Thank you very much. Fantastic performance. Thank you very much for that. I think you raised a few points and there are loads of questions to answer. I would like to start with questions. There is one here from the audience. It says, from your personal knowledge of testing AlexNet and LeanNet 5, which process will be best used for healthcare diagnosis and treatment, and why? Shall I repeat it?
No, I'm thinking about this question. Actually, this is a very good question. And in our experience, and so it's difficult to tell actually which network we should use. Depends on the applications. For example, if your classification is just black white, or the data are so distinct between different groups, I would use a simple model like AlexNet. But if the problem is more complicated, and there is only small margin of difference between two and the groups, for example, you want to classify, I would prefer complicated models like Inception V3 or even in ensemble approach.
Okay, thank you for that. Based on your experience, that's my question, personal question. Based on your experience, this artificial intelligence, how far do we have to wait for a kind of very extended use within the NHS? Where are we now if, say, AI is used, say, in 90% of the cases? Where are we now? If it's 100% would be the most. Where are we now at this moment in time? Can we say it's about a couple of percentage points or?
Yes, actually, AI now has been introduced into NHS, at least at some eye screening centers. They are going to test those AI platforms. Yeah, in terms of diabetic eye screening, yeah, this is in practice. And also recently, there are many products that have got FDA and CE marking. So we are expecting there are more products.
Right. So from your experience, personally yourself, to what extent would you trust AI approaches in a medical profession?
For me, and I trust AI very much now.
Very good then.
Because we have been working on different projects and validated and extensively. So
I'm pretty sure we need it. We need artificial intelligence. Yeah, we do need that. Okay, thank you very much for this contribution. There is one question here to perhaps Rachel. I know there were problems with surface wearing in hip replacements. Has the coating proved promising in these tests too?
So the application that we would like to use those coatings for is sort of, any sort of metallic implants. So either it can go for hip replacements, which are metallic, or it can go for dental implants. So we have proven it in our lab, where the coatings are quite stable and they're able to prevent bacterial adhesion, but they are happy enough, the osteoblast cells grow on them. But what we need to do is a more rigorous test. We need to test it within an animal model, sort of the next stage, to see if it's a viable coating for use in clinic.
So that's in the pipeline, in progress.
Yes.
Thank you so much, Rachel. There is one question which is perhaps, it's to me, it's not to the speakers. How do I get a CPD certificate for attending this webinar? Well, we should have started with announcement. So this webinar is worth 2 hours CPD. And anyone wanting to have a CPD for attending this one, perhaps can send an email to Ashkan or to me, and we are going to send you a CPD certificate. Thank you very much. I think that's all. Are there some more questions? I can't see any more questions.
No, I don't think so. I don't think so.
There is one question here. I don't know. Perhaps it was to Professor Ahmed. Which of the treatments mentioned will be most suited to treat damages caused by glaucoma disease? I don't know who answers this question. Who of you?
Sorry, I didn't get that. Can you repeat that, please?
Which of the treatments mentioned will be most suited to treat damages caused by glaucoma disease? That's the question.
Yeah. So this question is for me.
I don't know. Perhaps it's
okay. Well, I think so what I know is that so in glaucoma, if damage takes place, it cannot be repaired, unfortunately.
Yeah. All right.
So in glaucoma, usually you get an elevated internal eye pressure, and this pushes on the optic nerve head and causes the damage in basically the cables connecting the retinal cells to the brain. And if these get damaged, they cannot be restored.
Which means it's a kind of irreversible damage, isn't it?
Yes.
Irreversible, which is a shame. That's what it is. Now, to all, it's my sort of question to all speakers. I wonder if you have any connection with the Institute of Molecular and Clinical Ophthalmology in Basel, in Switzerland. No. They do a fantastic job. I got some connections. I can connect you to them. So it's Institute of Molecular and Clinical Ophthalmology. Ophthalmology, yeah, in Basel. Ashkan, back to you then.
Okay, thank you very much. So at the end, I just want to thank our speaker for excellent presentations and for accepting to do this talk tonight. And thank you, Andy and Rob McDonald and Sandra Lucy from IET for helping me organize this. So if speakers have anything to say at the end.
Well, I'd like to say a few more words, perhaps, on behalf of the IET. Yes, I just joined Ashkan, and thank you ever so much for spending your time, dedicating your time for this event. And it's a shame that we couldn't meet personally, but hopefully there will be a time when we can do it. And again, by the same token, I have to say thank you to the audience to attend this seminar. And I'm really sure that I wish they could attend. You could attend again in our next event, sometimes in February. Until then, I'd like to see you. Bye-bye now.
I think what I would like to add is that if anybody in the audience would like to get in touch with us and get involved in activities, please just drop us a message, and you can join lots of groups and activities that we have at IT. So thank you, everyone.
Thank you. Thanks a lot. Bye now.
Thank you.
Thanks. Bye.
Bye.